Dear students, I am Muzaffar Ahmad Shah, Senior Lecturer Botany, Boys Higher Secondary School, Ganderbal. Today, we shall have a discourse on a very important topic, that is pollen pistil interaction. There are two aspects of this pollen pistil interaction. One is precisely we shall be discussing the a simplified version of the molecular mechanism and the molecular interaction that is involved in it. And the second part is that you often study synonymously with the course of fertilization, right. So, today we shall be discussing the molecular interaction and simplified version of the molecular mechanism involved in the pollen pistil interaction, right. So, having said this, let us be specific that a pistil the stigmatic surface or the stigma of the pistil is sufficiently equipped to differentiate between the right and the wrong type of pollen. It will allow only the right type of a pollen to germinate and to proceed further towards an ovule or an embryo sac. This is one. Second, this stigmatic surface there is an on stigmatic surface and this upper part of the style, there is an array of reactions involved upon the landing of the stigma on the surface of this, upon the landing of the pollen grain on the surface of stigma. This is one. Second, now if this pistil carrying a functional gamete, carrying a functional embryo sac is unable to set seeds following a pollination by a viable and a fertile pollen grain, which may be able to fertilize the other pistil, but not the pistil in question. And then the two, this pollen grain and the pistil, these are said to be incompatible, and the phenomenon as sexual incompatibility. Now, there may be say two types of this sexual incompatibility. One, you say between individuals of different species, simply that you call interspecific incompatibility, and the second one is the incompatibility between individuals of the same species. This you call intraspecific incompatibility. Now, see, both are very significant. Now, this interspecific incompatibility, it defines the boundaries of a species and it allows the species to maintain its distinctness. Now, this intraspecific incompatibility very much significant, that it allow variation to a certain degree. Now, this intraspecific incompatibility, simply we call it also, that is precisely our topic today, that is self incompatibility. That means a plant with a functional pistil with a viable and having producing viable and fertile pollen grains, when pollinated, is not able to set seeds. I mean, that phenomena is precisely called self incompatibility. Now, without going into say different types and typifications of this self incompatibility, we shall be focusing on two different types of self incompatibility. I mean, that shall be the part of our discourse today. Now, first, depending on the origin of the factors precisely from the pollen side that decides the mating types, we have two different types of self incompatibility. One you precisely called gametophytic self incompatibility, and second you call it GSI system, GSI, gametophytic self incompatibility, and second you call sporophytic self incompatibility. Now see, gametophytic self incompatibility and the second is sporophytic self incompatibility. Now see, prior to going into the discussions of both these and the molecular mechanism involved, may I remind you the fact that most of the plants in nature are outbreeders. That means there are devices, there are modifications, contrivances which largely favor outbreeding, that is cross pollination. Say different modifications are there, maybe the say spatial difference in the presentation of the pollen grain and its receptivity and its reception, maybe the time difference that you simply call the dichogamy, or previously you call the previous one you call the hercogamy. There is another mechanism you call unisexuality involved which favors outbreeding, and the different morphotypes involved largely in heterostyly. But among all, the most widespread and the most effective barrier that favors outbreeding, that adds to the variation which is inevitable in outbreeding and largely means important also, that is self incompatibility. Now, so coming back to this gametophytic self incompatibility and sporophytic self incompatibility. Now, gametophytic self incompatibility is the one that is decided by, that is determined by the genotype of the pollen grain. Now, this gametophytic self incompatibility is decided by the genotype of a male gametophyte. Pollen grain. So, genotype of the male gametophyte. Okay. Now, and the sporophytic one is the genotype of sporophytic tissue sporophytic tissue of the plant from where pollen grain is derived. Okay. I mean the contribution of largely of say tapetum and the anther locule, genotype of the sporophytic tissue of the plant from which pollen grain is derived. So, having said this, now we shall simply now discuss the molecular mechanism separately in gametophytic self incompatibility and then sporophytic self incompatibility. Now, see, I will try to have a simplified version of the molecular mechanism involved, though it is very complex. So, first of all, we shall discuss the pollen pistil interaction, the mechanism involved in gametophytic self incompatibility, that you call GSI system. Now, simply pollen grain gets alighted on the stigma, simply pollen grain lands on stigma, then simply because of the say exudation on the surface of stigma, that results in the pollen grain germination, right. Now, say now pollen grain germination, that means results in the formation of the pollen tube, okay, pollen tube formation. Okay. Now, this pollen tube, it passes through the stigmatic surface and then reaches style. Now, say in case of GSI system, now here is a catch in say style. Now, in this after pollen grain germination, it pollen tube, we write say flow chart, I am just showing it with the help of a flow chart to make it more simple. Now, this pollen tube, its entry, pollen tube entry into style. Okay. Now, pollen tube entry into style. Now, here what happens? Now there is a recognition reaction taking place between the stylar receptor proteins here, the stylar receptors, or you call stylar receptor proteins recognize the marker proteins on the surface of the pollen tube. Stylar receptors recognize after pollen tube entry into style, here stylar receptor proteins recognize pollen tube marker proteins of pollen tube, right. Now, thereafter, upon the interaction of the markers and the receptors, the stylar cells produce a gene regulated, that is a gene responsible for simply self incompatibility, that you call, that is highly morphic gene, the single gene locus, highly morphic single gene locus, that you call simply S locus, is highly polymorphic. Okay. The self incompatibility gene, that is S gene, is highly polymorphic with different number of alleles, I mean that you call S locus, and that is a typical example of multiple allelism, that is self incompatibility gene. This S locus is a typical example of self incompatibility in plants, right. So, what happens, see, after this receptor marker interaction here in the stillar itself, the stillar cells, the stillar canal cells that produce S specified RNases. Upon the interaction, the stillar cells produce S RNases enzymes that bring about the degradation of RNA, right. Now, say this say after interaction, this say then subsequent production of the sRNAs by the stillar canal cells, it then moves into the cytoplasm of the pollen tube, right. This sRNAs, this now moves into pollen tube, right. Now, here, now there are two fates depending on the type of the sRNAs. There are two possible fates of pollen tube. One, that if it is this RNAs upon arriving in the cytoplasm of the pollen tube, if it is self type, that means if it is genetically related, I mean simply self incompatibility decides the genetic relatedness. If it is self type, that means genetically related, then what happens, this sRNA, it is allowed to survive, means it remain as such in the cytoplasm in the or in pollen tube. See, it is allowed to survive, this RNA is allowed to survive and how this RNA affects the pollen tube, I mean precise manner is unclear, but the degradation of the RNA seems to be a plausible mechanism here simply, but we are not I mean certain about that, if the degradation of RNA is a causal effect or is the end effect, but largely it has been proven that is a plausible mechanism, that the sRNAs which remains intact in the pollen tube, it brings about the degradation of rRNA. So, I mean in case of self type RNAs, it is allowed to survive in the pollen tube, inside the pollen tube. That means when it is allowed to survive, it brings about the degradation of rRNA, right. So, as a result of this degradation of rRNA, what happens? Simply translation, I will show it here again. Okay. If it is allowed to degrade, sorry, now it results in the halt of translation, simply halts, comes to an end because of the degradation of ribosomal RNA. And then when translation halts, simply after that pollen tube growth stops altogether. This pollen tube growth stops altogether in the style itself, then it may not be able to, when it stops, the cessation of the pollen tube occurs, then there is no possibility that it may reach into an ovule or an embryo sac and may bring an effective fertilization and seed set. So, no fertilization and consequently no seed set, right. So, this was the first fate of this the interaction in the setilar canal cells between the receptor proteins and the marker proteins, right. So, one possible fate is that the sRNAs may diffuse into pollen tube, and which being the self type, being the genetically related, it may be allowed to survive. So, as a result of that, it may bring about the degradation, altogether degradation of the RNA, and then consequent say stopping of or the say translation halts altogether, then pollen tube growth stops altogether, then that means no further passage, no delivery of the gametes, no fertilization and no seed set. This is one possible fate. The other is being the, if it is this sRNAs if it is non self type, if it is non self type, that means if it is genetically unrelated. Okay. Now, upon its entry into a pollen tube, one important mechanism that this RNAs upon its entry into a pollen tube simply comes into play very highly regulated mechanism, that is this sRNAs is being destroyed by the if it is non self type, it is destroyed by the ubiquitin, means a protein mediated proteasomal pathway. That means ubiquitin protein along with the other proteins in a complex system that you call the proteasomal pathway, it brings about the destruction of this sRNAs, right. So, after that simply that means that results in the degradation of the sRNAs itself, destroyed by the ubiquitin mediated proteasomal pathway, right. Then that means then growth of no effect means growth of pollen tube continues. When this sRNAs by this mediated pathway gets destroyed, gets degraded, no effect on its ribosome, this RNA content, mRNA or otherwise rRNA, then as a result of that the growth of the pollen tube continues, okay, that results in the its entry into an ovule, into an embryo sac, then delivery of the these male gametes and resultant fertilization and consequent seed set, right. So, beta, in case of GSI system, where there is an interaction between the genotype of the gametophyte and the stellar canal cells, we see the recognition reaction and the rejection reaction both taking place in the style. The reaction, the recognition, that gets unnoticed on the stigmatic surface, but on its arrival in the style, the mechanism comes into play. The pollen tube or the genotype, or the say simple genotype of the male gametophyte or pollen tube itself, it gets recognized there and rejected there. So, in case of GSI system, we have the recognition and rejection reactions taking place in style itself, right. So, now after this GSI system, we would like to have a comprehensive understanding of SSI system, right. So, for that we shall have to take a little pause, we will come back and we will continue with the molecular mechanism of self incompatibility in sporophytic self incompatibility.